Bismillahirrahmanirrahim. Dear all students, welcome to the new lecture of Histo techniques. Today we will discuss about the mechanism of fixation, that how different type of fixation, different type of fixative, they fixes the tissue in the structure in which they are literally present within the body. In today's lecture we will discuss about the different precautions during the process of fixation, and then we will discuss the mechanism of fixation of different fixatives, and in the last we will discuss what are the different factors that have effect on the process of fixation. The precautions in fixation. So the first one is that the tissue, it should be free from every type of excessive blood before putting in fixation, because this blood that is coagulated after putting in fixative, and then it hinders the other processes during the tissue processing. So therefore, before putting any tissue into the fixative, it should be cleared from the blood which is present in that tissue. The second thing is that before putting the tissue in a fixative, if it is thicker than 5 millimeter or 10 millimeter, then it should be sliced into thin sections, and these slices should not be thick more than 5 millimeter. It should be round about 3 to 5 millimeter thickness. And before putting any tissue in a fixative, it should be noted that the quantity of fixative should be more than the tissue, because if tissue is not properly immersed in the fluid, then the area, the portion which is not immersed in the fluid, so that is not fixed, and then it can hurdle the results, and we cannot properly examine that area which is under fixed or that is not properly processed. So therefore, the amount of fixative should be at least 20 times more than the volume of the tissue. And the last precaution, that the tissue with fixative should be in a tightly screw capped bottle, because some of the fixative, they are volatile, so therefore the container should be screw capped. So these are the precautions which need to be followed during the process of fixation. The mechanism of fixation, how different type of fixative they fixes the tissue, what is the science behind this fixation. So there are two methods by which the fixative move their work. The first one is the dehydration and coagulation of protein, and the second one is the cross linking of different bonds. So these are the two mechanisms on which different type of fixative which are routinely used in the histopathology, they perform the process of fixation. The first one is dehydration and coagulation of proteins. In this method, methanol and ethanol are commonly used fixative, and they work as a dehydrating agent and coagulation of protein. In the in methanol and ethanol, these are the fixative and they coagulate the tertiary structure of protein by the process of dehydration and then destabilizes the hydrogen bonds that is present within the different proteins. But the secondary structure of protein that is maintained, that is not disturbed. So therefore, overall the structure of the tissue is maintained by fixation in the methanol or ethanol, and among these two, the ethanol is relatively stronger dehydrating agent. So ethanol is commonly used, and the concentration of these two alcohols is 60% for the ethanol and 80% methanol. So methanol is used as 80% concentration and ethanol is used in 60% concentration for the process of fixation. This is the picture which shows that this is a tissue and the water molecules inside the tissue. So when alcohol is put into the solution or tissue is immersed in alcohol for fixation, this alcohol attaches and removes the water molecule from the tissue and dehydrates the tissue and coagulates the protein. But there are certain disadvantages of dehydration and coagulation of protein mode of action of fixative. The first one is that it causes the shrinkage of the cells, and the second one, removal of the soluble substances from the tissue. So when larger quantity of water is removed from the cells, then the size of the cell it is decreased and causes shrinkage of the cell as well as the whole tissue. And the second thing, that the removal of soluble substances from the tissue, certain soluble substances they are present in the tissue. So when these soluble substances are removed from the tissue, then the certain type of stains they cannot be applied to these tissues. So therefore, these are the two disadvantages of the alcohol fixatives. The second type of mechanism of fixation, that is the cross-linking. So these are the different type of fixatives which have this type of mode of action. The first one is formaldehyde. Formaldehyde reacts with different side chains of the proteins, and after this reaction they form a hydroxymethyl side group. These compound when formed, they are highly reactive and they cross links with each other by forming a methylene bridge. So when this methylene bridging is started, then slowly and gradually there is a cross linking between the intermolecular and intramolecular structures within the tissue. This formation of cross linking between the molecules of the tissue, they form an insoluble product, and this insoluble product, this act as a tissue, and the end product is the insoluble product. The second type of fixative which works as a cross-linking mechanism. So glutaraldehyde is the second one, and this glutaraldehyde have two aldehyde groups. These aldehyde groups are separated by 3 methylene bridges. These aldehyde groups react with the amino group of the amino acid lysine. So the other free aldehyde group, they help to cross link. This cross linking is rapidly and irreversibly within the protein and form a product. The penetration of glutaraldehyde is slightly slower than the formaldehyde. That's why formaldehyde is preferred as compared to glutaraldehyde. The third fixative, that is osmium tetroxide, which also work as a cross-linking fixative. This type of fixative is used alone or in combination with the other fixatives, and mostly this osmium tetroxide is used for the electron microscopy of the tissues. The osmium tetroxide, it causes the oxidation of unsaturated bonds in the lipids, and this oxidation converts the unsaturated fatty acid into a stable product that is called glycol osmate, and the osmium tetroxide may react with the two unsaturated carbon atom of the lipids and may cross links. This is the mode of action of formaldehyde. The formaldehyde molecules, it react with the proteins, and this is the cross linking of the different types of proteins. And here the osmium tetroxide, it reacts with the lipids and cross links the lipids with each other, forming glycol osmate. So these are the mode of action of formaldehyde and the osmium tetroxide. The factors which affect the process of fixation. There are different factors which have effect on the process of fixation. The first one of them is the pH of the fixative. The second one is the temperature of the fixative. Third one is the duration of fixation, the time for the fixation. The fourth one is the osmolarity of the fixative solution, and the concentration of the fixative solution, and the agitation of the solution. So these are the factors which have effect on the process of fixation. So let's discuss the pH of fixative, how this pH of fixative have effect on the process of fixation. The fixative, it works better when it have a neutral pH, and the good fixation, it occurs when the pH is in between 6 and 8, because when the fixative had this neutral pH, then it causes no morphological changes or distortion, and the tissue remains in the normal and near to the original condition. The changes occurs in the ultrastructure when there is a change in the pH, that is, the pH is too low or it is too high. So therefore the pH should be within the neutral range, and the buffers should not react with the fixatives. That should be a point, that if a pH of the fixative is maintained between 6 to 8 degrees centigrade with the help of buffers, so these buffers should not react with the fixative. The commonly used buffer in the fixatives are the phosphate buffer, the bicarbonate buffer, tris buffer, and acetate buffer. The second factor that which affects the process of fixation is the temperature. For the fixation, room temperature is suitable during the routine processing, routine fixation of the tissues, and when the temperature of the fixative is increased, then this higher temperature it causes in the reduction of the time of fixation. For example, if the temperature is increased for up to 60 to 65 degrees centigrade, so on this temperature the fixation time of the tissue is decreased by increasing the temperature, and if the temperature is very much increased, then this destroys, this affects the antigens which is present on the tissues or within the tissues. So therefore higher temperature should be avoided, and in contrast to highest temperature, the enzymes they are better preserved in the lower temperature of the fixative. If enzyme histochemistry is to be performed on the tissue, then for that purpose 0 to 4 degrees centigrade is the most suitable temperature, and this temperature better preserve enzymes in the higher temperature. The duration of fixation is the next factor which affects the process of fixation. The depth of penetration of fixative is directly proportional to the square root of the time of fixation. If the penetration rate of formalin solution is 1 millimeter per hour, so the presence of blood that hinders the penetration of the fixative, and it is preferable to wash the tissue specimen before putting it into the fixative, because the blood that hinders the penetration power of the fixative, and if there is a thick tissue, then this tissue should be sections as 3 to 5 millimeter, and overall formalin fixes tissue within 24 hours. The next factor which affects the process of fixation is the osmolarity of the fixative solution. If the fixative solution is hypertonic, then this solution causes the shrinkage of the tissues, and if there is hypotonic solution, then this hypotonic solution induces or activates and causes the swelling of the cells. Electrolytes or normal saline or sucrose, it is added to fixative to maintain the osmolarity of the fixative solution. So if there is a hypotonic solution, then electrolytes are added to it, and mildly hypertonic solution, that is 400 to 450 milliosmolar solution, is preferable for usage and fixation of tissues in routine histopathological laboratory. The concentration of fixation, the concentration of fixative, it is directly proportional to the fixation. If the concentration of the fixative is high, then the time of fixation and the process of fixation, it is the time is decreased and the process of fixation that is increased, and if the concentration of fixative is decreased, then the process of fixation is decreased and the time required for the fixation of the tissue that is high. So higher the concentration of fixative, This may causes hardening of the tissue and causes shrinkage and other artifactual changes. But the mildly lower concentration of fixation with neutral pH, it is required for the proper fixation of the tissues. That is why the optimal concentration for formaldehyde or formalin is 10%, and in the routine 10% formalin, buffered formalin, is used for the fixation of the tissues. That was all for the today's topic. Hope you have learned about the precautions during the process of fixation, the mechanism of fixation, and the factors which affect the process of fixation. For further study, you can look through these books regarding the topics which are discussed today. Thank you for watching. If you have any question, you can ask through your question box. Thank you. Assalamu alaikum.